Hello everyone, and welcome to today's webinar, Unveil the Immune Response to Cancer and Infectious Disease with Powerful Immunoassays. I am Jennifer Woods of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing the presentation. This presentation is educational and thus offers continuing education credits. Please follow the process to obtain your credits by clicking on the Continuing Education Credits window. I'd like to now welcome our speaker, Sigrun Badrnya, Product Development Manager, Thermo Fisher Scientific. Sigrun, you may now begin your presentation.
So thank you very much for the introduction, and again, welcome everyone to this webinar, which will provide insights into assay solutions to explore major questions around the host immune response to cancer and infectious disease. So unraveling the interactions between the immune system and cancer cells or pathogens such as viruses is key to understand and potentially control both cancer and infectious disease. As depicted in the figure on this slide, the immune system is an extensive network involving numerous cells and molecules, including a large number of cytokines and chemokines that shape local and systemic immune responses. There's evidence for a dual role of inflammation, also in the context of a cancer, involving several cytokines, which are either cancer-promoting or rather support anti-cancer responses and treatments, in conjunction with soluble receptors and the ligands of T cells and innate immune cells, including natural killer cells, as summarized in the figure on the right, that are critical players in immuno-oncology, which rapidly emerges as a research field focusing on innovative therapeutic approaches that take advantage of an individual's immune system to fight a cancer. So a hyperinflammatory state of the immune system, also known as a cytokine storm or cytokine release syndrome, has not only been described as a possible side effect of cancer immunotherapies, but also as a common complication of various other pathological conditions, including infection by the novel severe acute respiratory syndrome coronavirus SARS-CoV-2. Such an overactive inflammatory response can cause collateral tissue damage as the immune system tries to expel the virus by recruitment and infiltration of immune cells into infected tissue. So high levels of inflammatory soluble factors, including interferons, interleukins, colony-stimulating factors, or tumor necrosis factors, as outlined in the table, can contribute to the symptoms of SARS-CoV-2 infection that might range from mild to more severe respiratory or even life-threatening conditions with dysfunction of multiple organs. So in this state, immunosuppression by selective blockade of cytokines is likely to be beneficial. Thus, there's much interest to uncover the complex interplay between soluble mediators implicated in the cytokine storm and to further elucidate the dynamic changes and interactions between the immune system, tumor cells, and viruses such as SARS-CoV-2. So one way to better understand differences in the immune responses of subjects is to look at biomarkers. Apart from soluble factors and secreted proteins such as cytokines and chemokines as mentioned, these can include immunoglobulins, immune cells involved, intracellular proteins that play a role in immune signaling pathways, or specific receptors and surface proteins that are of particular relevance in immuno-oncology, or key entry targets for SARS-CoV-2, such as the angiotensin-converting enzyme 2, ACE2, that serves as a host cell receptor. So finally, analyzing all these biomarkers and soluble factors of the immune system will help to identify differences in cytokine repertoires and to characterize changes in the pattern of proteins and other target analytes in order to relate them to specific mechanisms underlying pathological conditions. For research purposes, the extensive portfolio of Invitrogen branded immunoassays meets a wide range of needs. So from build it yourself immunoassays by choosing from our large selection of antibodies, proteins, and other reagents, to ELISA kits for single target protein measurement, including coated yourself ELISA kits with matched antibody pairs as an affordable option for less stringent requirements, to validated ready-to-use ELISA kits, and newer platforms like ProQuantum, which are highly sensitive, homogeneous assays with low sample consumption, using proximity-based qPCR amplification for protein target quantitation on any PCR instrument. And finally, multiplexing solutions on Luminex-based systems for measuring multiple proteins or RNAs simultaneously in one well. In this webinar, I will particularly focus on immunoassays in the ELISA format, as well as on multiplexing solutions that have been specifically designed to support both immuno-oncology and SARS-CoV-2 research. So with over 700 targets, Invitrogen ELISA kits are a cornerstone to measure cytokines involved in pro-inflammatory responses, also for SARS-CoV-2 research. In a recent publication as cited here, ELISA kits were used to measure several cytokines, including IL-17A, TNF-alpha, or IL-6, to better understand immune dysregulation of pneumonia induced by SARS-CoV-2. In this case, a unique signature of the soluble mediators could be indeed identified in subjects infected with SARS-CoV-2 in association with a macrophage activation syndrome and defective antigen presentation driven by IL-6, which could be in part rescued by addition of a specific IL-6 receptor blocker. For highly multiplexed targeted gene and protein expression studies, Invitrogen assays make use of the multi-analyte profiling technology that is compatible with any Luminex instrument to provide a flexible, high-throughput solution in a quantitative format. So with the incorporated branch DNA technology, QuantiGene Plex assays allow for accurate multiplex gene expression profiling of up to 80 targets per well directly from a blood, tissue homogenate or lyse sample without the need for RNA purification. While ProcartaPlex immunoassays for protein quantitation enable the simultaneous detection of up to 65 targets in a single sample of plasma, serum, cell culture, supernatant, and other bodily fluids, requiring only 25 to 50 microliter of sample material, thus combining the efficiency of multiplexing with the accuracy, sensitivity, and reproducibility of a traditional ELISA. In fact, one instrument platform enables analysis of multiple RNA or protein targets in the sample of interest, and thereby providing a comprehensive analysis solution for transcriptional and the proteomic level. Importantly, these multiplex assays may be customized by either selecting any RNA transcript or by choosing from a portfolio of more than 300 protein targets from human, mouse, and other species, covering cytokines, chemokines, growth factors, but also coagulation factors, immunoglobulins, biomarkers for immuno-oncology or SARS-CoV-2, and many other areas of biology and research. The multi-analyte profiling technology is basically based on proprietary MUCplex and microplex microspheres, so-called beads, with a magnetic core and polystyrene surface that are internally color-coded, resulting in 100 unique spectrally distinct sets or bead regions that can be individually identified by one of the Luminex systems. So each bead set is assigned to a different analyte and conjugation of a target-specific antibody to a distinct bead set for protein quantitation, or using target-specific probes to capture RNAs, finally allows analysis of multiple analytes, and in essence gives rise to up to 100 different assays in a single well. The two instrument lasers are one laser to excite the bead dye and one green laser to excite the phycoerythrin detection dye, enable classification of the respective bead region and quantitation of the target analyte in parallel. So how can this technology and platform assist SARS-CoV-2 research? On the ProcartaPlex platform, a curated panel for cytokine storm research combining 21 selected and highly relevant targets for inflammatory cytokine profiling is available, in addition to pre-configured panels for immune monitoring, and besides customized combinations of available targets using our mix and match offering to design a unique panel for SARS-CoV-2 research appropriate to your needs. To further characterize the host immune response to SARS-CoV-2 on a transcriptional level, the targets for cytokine storm research are also available on the Quantigene platform, and there's highly customizable assays. QuantiGene Plex can incorporate host and virus targets in the same panel, allowing the relative gene expression data to be obtained for these targets from the same sample. Custom research panels on this platform can include up to 49 targets relevant for studying SARS-CoV-2 infection. So by combining immune-related biomarkers with targets relevant for the viral infection process, this gene expression analysis can help to compare the transcriptional response and to better understand the molecular basis that may underlie SARS-CoV-2 induced infection. In collaboration with Professor Angela Slitt from the University of Rhode Island, the performance of a QuantiGene Singleplex assay for detection of SARS-CoV-2 viral RNA was evaluated for research purposes. So for this purpose, 3 different RNA samples extracted from nasopharyngeal swabs that were provided by the Rhode Island Department of Health with known CT values from qPCR testing, including a sample with low, high and very high positive qPCR signal together with the reference material were tested. To showcase linearity, the estimated copy number was graphed against the relative light unit signal, demonstrating an excellent performance with an R-square of 0.98 corresponding to qPCR results. So an aspect that is of particular interest also in this research field is the humoral immune response to SARS-CoV-2. As summarized in this figure, actually 4 main structural proteins have been identified that are implicated in SARS-CoV-2 infection and replication, including the envelope, the membrane, the nucleocapsid, and the most prominent spike or S protein, which mediates the viral entry into host cells by interacting with ACE2 as a receptor on the host cell. Recent evidence also suggests additional factors to potentially facilitate the viral host cell entry, which points out that there are still mechanisms that need to be further explored in more detail in this dynamic research field. So as the virus enters the host cell and following replication, release and subsequent ingestion by antigen-presenting cells, fragmented viral antigens are presented to T cells, which in turn interact with and activate B cells that finally secrete SARS-CoV-2 specific antibodies of different classes, including IgM, IgA, or IgG. A certain portion of these antibodies is capable to neutralize specific viral antigens, thereby blocking their interaction with the respective host cell receptor and potentially prevent SARS-CoV-2 infection. So understanding of the antibody-mediated adaptive immune response to SARS-CoV-2 is key not only to define testing strategies, but also in view of the development of vaccines and other future treatment options, and will require reliable serological tests. In addition to other powerful technologies and tools for SARS-CoV-2 research, Serological assays will allow to evaluate the seroprevalence, to monitor dynamics of antibody titers over time, and to finally determine the extent and duration of potential immunity in subjects following SARS-CoV-2 infection or vaccination. To assist researchers conducting epidemiological or other research studies, our portfolio of SARS-CoV-2 research tools is also complemented by serological immunoassays to finally provide a comprehensive solution for exploring host immune responses at each stage during the course of viral infection. So besides profiling of cytokines related to the cytokine storm and analysis of viral RNA and other target analytes on the transcriptional level, our serological immunoassays will allow accurate detection of SARS-CoV-2 specific antibodies and further characterization of their neutralizing capacity. In this regard, I would like to introduce two new serological immunoassays for research purposes, a multiplex ProcartaPlex panel, as well as one of our available assays for SARS-CoV-2 research in the ELISA format. The human SARS-CoV-2 spike Ig total ELISA combines the spike trimeric structure immobilized to the surface and total antibody detection with the flexibility of either qualitative or quantitative readouts, providing one of the best platforms for sensitive and specific detection of SARS-CoV-2 antibodies. Taking a look at the workflow of this assay, which uses a spike trimeric protein as specific viral antigen coated onto the surface of a 96-well plate to bind total antibodies to SARS-CoV-2 present in a blood plasma or serum sample. These bound complexes are then detected by a horseradish peroxidase conjugated antibody, and following an incubation step with a substrate finally results in a colorimetric readout with a short time to result of only 1 hour and 15 minutes. Evaluation of the performance of this ELISA included 39 PCR-confirmed SARS-CoV-2 positive samples from convalescent subjects and 160 PCR-negative control samples, as shown by the two bar charts here on this slide. 2 samples from a subject declared PCR positive were identified as negative with detected signals below the set cutoff as illustrated in the upper bar chart. However, these samples were confirmed to be negative also in other serological assays that were performed, overall underlining the excellent specificity and sensitivity of this ELISA. A further screening with control samples positive for other respiratory viruses, including influenza A, B, or parainfluenza strains, did not show any cross-reactivity with the spike trimeric protein. As depicted in the graph, a high control indicated in red, a calibrator to define the cutoff in yellow, and a low control indicated in green are also provided as part of the ELISA kit. Except for 3 PCR positive samples here indicated in orange that could be reconfirmed, all tested control samples here in blue were below the set cutoff, finally resulting in 100% specificity of the assay. Benchmark testing of the Invitrogen branded ELISA to an assay of another supplier currently on the market showed some discrepant results for certain samples as indicated by the red arrows in the 2 graphs. So one healthy negative sample that was identified as borderline in the Invitrogen assay indicated in green in the upper bar chart, however, was found to be above the set cutoff and thus identified as false positive in the competitor assay as presented in the bar chart below. Furthermore, 3 PCR confirmed positive samples that could be verified in the Invitrogen ELISA were found to be borderline or clearly below the cutoff level and thus identified as false negative in the competitor assay. Overall, the Invitrogen ELISA shows more samples with results as expected and therefore outperforms the other suppliers' assay regarding specificity and sensitivity. So besides the SARS-CoV-2 IG Total ELISA, the new serology multiplex panel on our ProcartaPlex platform provides a comprehensive screening tool to capture antibodies to more than one SARS-CoV-2 specific antigen and allows to distinguish antibodies to these subsets of SARS-CoV-2 as well as to other related coronavirus strains while saving time and also resources. A solid knowledge of the SARS-CoV-2 protein structure and homology to other coronavirus strains is crucial to understand how the virus acts and also affects the host immune system. So while the N or nucleocapsid protein primarily regulates viral RNA synthesis, the spike or S protein plays a critical role in mediating fusion and viral entry into the host cell as mentioned. So as illustrated by the three-dimensional schematic drawing of the structure, the S or spike protein assembles into a trimer to finally form the distinctive crown-like appearance and comprises multiple parts, an N-terminal S1 subunit, including the receptor binding domain that specifically recognizes the host ACE2 receptor protein, and a C-terminal S2 membrane fusion subunit. As summarized in the table, N and S proteins show varying degrees of homology between the other 6 strains of coronaviruses, while the N protein shares about 90% amino acid identity with the previous SARS coronavirus, SARS-CoV-1, and about 50% with the Middle East respiratory syndrome related coronavirus. The S protein shows less structural similarity to other coronavirus strains, which is particularly low for both the N and the S protein between SARS-CoV-2 and the 4 listed endemic coronavirus strains, which are also called common cold coronaviruses. Taking a closer look at the design of this 11-plex panel, which enables the detection of total antibodies against 4 different SARS-CoV-2 antigens, including the nucleocapsid protein, the spike protein, and the subdomains that are considered as the most immunogenic, as well as to the S1 subunit of 6 other coronavirus strains next to a negative control. So this broad epitope coverage is a unique and integral part of this serological multiplex assay, which provides a more complete antibody profile and consequently increases the sensitivity and the likelihood of predicting immunity status. Among various spike protein domains, the S1 subunit shows the lowest homology between these coronavirus strains, which makes it more specific to SARS-CoV-2 antibody detection and allows to differentiate between coronavirus strains, including the 4 endemic strains of common cold coronaviruses that are quite prevalent in the general population, however, cause only mild respiratory symptoms. So the recent evidence supports the hypothesis that pre-infection with these common cold coronaviruses may provide immunity also against SARS-CoV-2. In this regard, a pre-existing T-cell mediated immune reactivity to SARS-CoV-2 that has been observed to some extent in the studied population and is supposed to be caused by prior infection with these common cold coronaviruses is discussed. This is quite an interesting finding, which might have also implications in view of vaccine development and should therefore be considered, and these common cold coronaviruses should be further monitored. However, these findings need to be confirmed and also more evidence provided to have more thorough evidence. The human coronavirus IG Total ProcartaPlex panel was evaluated by Dr. Doug Fraser at the Western University and Lawson Health Research Institute in stratifying studies of intensive care unit subjects with SARS-CoV-2 antibodies. 14 healthy controls, 14 SARS-CoV-2 infected ICU subjects, and 14 non-SARS-CoV-2 infected ICU subjects were included, and blood plasma samples profiled at different time points. So as illustrated in the box plots, the mean fluorescence intensity signals for immunoglobulins against 4 different SARS-CoV-2 antigens demonstrate robust immune responses over time, with a significant increase in SARS-CoV-2 immunoglobulins in SARS-CoV-2 infected ICU subjects, here indicated in red, as compared to healthy controls and non-SARS-CoV-2 infected ICU subjects already at ICU day 2, and even more pronounced from ICU day 3 on. Analysis of the complete plasma SARS-CoV-2 antigen responses using a state-of-the-art machine learning algorithm, as depicted in the TSNE plots on the left, revealed a distinct discrimination between healthy controls, here indicated in green, and SARS-CoV-2 positive ICU subjects, indicated in red, with increasing classification accuracy over time that reached 100% on ICU day 3. Table D lists the relative importance of each of the SARS-CoV-2 antigens between the groups, showing a predominant, however, equal contribution of the spike protein and its subdomains as compared to the nucleocapsid protein. Similar results were observed when comparing complete plasma SARS-CoV-2 antigen responses between SARS-CoV-2 positive ICU subjects and non-SARS-CoV-2 infected ICU subjects on respective days. Similarly, the plots on the right demonstrate a clear differentiation and stratification of these two groups of critically ill subjects, further underlining the excellent performance of this multiplex assay regarding specificity and sensitivity. So apart from SARS-CoV-2 research, powerful immunoassays designed for targeted approaches as well as for broad immune profiling of multiple biomarkers are also available to further elucidate the interaction between the immune system, the tumor and tumor environment, and to explore all facets of immune checkpoint pathways and support cancer biomarker discovery and verification across key immuno-oncology research areas. So among the key research areas in immuno-oncology, cancer vaccines have evolved as a strategy to stimulate a strong immune response and to support a robust antigen presentation based on antigens and neoantigens released by cancer cells upon cell death, besides the chimeric antigen receptor T cell, or abbreviated CAR T cell therapy, and immune checkpoint inhibition that are two of the most promising and rapidly emerging approaches. So while CAR T cell therapy further expands the adoptive cell therapy or transfer, relying on the reinfusion of a subject's T cells after specific genetic modification, which finally enables them to recognize and destroy cancer cells, the approach of immune checkpoint inhibition is based on therapeutic antibodies targeting immune checkpoint pathways as a novel way to attack tumor cells. Immune checkpoint pathways are critical in balancing immune responses in order to protect tissues from damage when the immune system is activated. However, cancer cells can dysregulate these immune checkpoints and through ligand receptor interactions use them as mechanism to ultimately evade immune surveillance. As pointed out by the figure on this slide, among the first immunotherapies to market were targeted to blocking the effects of cytotoxic T lymphocyte antigen 4, CTLA4, and programmed cell death 1, PD1, with several therapeutic antibodies currently approved as immunotherapeutic drugs. CTLA4 plays a role during the priming of T cells by antigen-presenting cells, and upon binding to its ligand B7 acts as a negative regulator of T cell activation and proliferation. Blockade of the CTLA4 receptor by specific antibodies, however, can boost T cell activation and thereby drive an anti-cancer immune response. The immune checkpoint protein PD1 is a regulator of apoptosis and critical for maintaining T cell expansion during the immune response. Binding of PD1 by its ligand PD-L1 expressed by a cancer cell can mask cancer cells from T cell recognition and finally attenuate immune responses. Since the discovery of cancer therapy based on inhibition of negative immune regulation via CTLA4 and PD1, that has also been recognized with the Nobel Prize for Medicine or Physiology in 2018. Many strategies and agents have been developed to target these immune checkpoint markers, which are actually in clinical trials or used in combination therapies that are promising to enhance anti-tumor immunity. This has also encouraged the research field to further investigate immune checkpoint pathways to potentially identify new drug targets also beyond PD-1 and CTLA-4. So in recent years, a main focus of the development of ProcartaPlex assays was on immuno-oncology biomarkers for investigating immune checkpoint pathway functionality in effector T cells and natural killer cells, which are involved in the regulation of both adaptive and innate immunity. Taking a look at the cancer immunity cycle on the left, which we are probably familiar with, schematically showing in a 7-step process how the immune system combats cancer cells, initiated by the release of cancer cell antigens upon cancer cell death, followed by the uptake and subsequent presentation of cancer antigens, T cell activation in the lymph nodes, the trafficking of activated T cell to the tumor site with infiltration into the tumor, and finally, the recognition and killing of the cancer cells with release of antigens that further drives this progression of the cycle. Each of these 7 steps is tightly regulated and involves the coordination of a multitude of checkpoint molecules and other cell surface receptors, as well as soluble factors such as cytokines and chemokines. So for many of these factors, we already provide multiplex panels, such as for pro and anti-inflammatory cytokines, which regulate step 2. And we have a complete portfolio of chemokines and adhesion molecules, which regulate T cell trafficking and infiltration. So our immuno-oncology checkpoint biomarker panels particularly focus on the steps 3 and 7, T cell activation and the killing of cancer cells, including the most important co-stimulatory and co-inhibitory molecules, which are often engaged by cancer cells to alter activation pathways and ultimately attenuate immune responses. And thus, these are also relevant targets for immune checkpoint inhibitors. Similar regulatory processes and checkpoints can also be found during innate immune cell activation, including natural killer cells, as depicted in the graph on the right. So a lot of receptor-ligand interactions are to be explored here, as well as potential therapeutic targets that might mediate anti-cancer immune responses, in addition to the important role of the adaptive immunity. This also further points out that immune checkpoint molecules are primarily recognized and considered as transmembrane proteins acting as receptor-ligand pair. However, soluble isoforms and shed variants are described for the majority of these molecules that can function either as immune adjuvants or decoy receptors and thereby regulate checkpoint pathways. This might, of course, have an impact on the clinical efficacy of individual checkpoint modulator drugs, and thus the immuno-oncology checkpoint biomarker ProcartaPlex panels have been specifically designed and optimized for the measurement of key immuno-oncology analytes in human or mouse serum, plasma, and cell culture supernatant samples. So as illustrated by the bar chart, a screen of more than 70 human melanoma samples finally proved and confirmed that all these soluble forms of checkpoint molecules included in the immuno-oncology checkpoint panel 1 and 2, which are 2 predefined panels, each including 14 targets, are indeed detectable in serum. Further evaluation of the immuno-oncology checkpoint panel 1 revealed that expression patterns of these soluble markers are dependent on disease state and cancer treatment. So as demonstrated by the 2 graphs, a reduction of PD-1 levels was detected in melanoma samples comparing early treatment to pre-treatment. And although we have in this case no further details and insights into treatment and respective responses, the results further highlight that monitoring of these soluble checkpoint molecules might provide valuable information in relation to the clinical outcome of therapy for early evaluation of treatment responses. And such characteristic responder profiles might therefore help to pre-select and also stratify subjects who may benefit from certain treatment. Moreover, concentrations of soluble checkpoint markers could also be determined in human breast cancer samples before and after chemotherapy using multiplex immuno-oncology checkpoint panels, as already published in 2016. There's evidence that chemotherapy-induced tumor cell death is associated with the release of intracellular molecules such as the cell death marker high mobility box group protein 1, HMGB1, into the microenvironment. So notably, soluble CD27, soluble CD80, and PDL2 correlated with the HMGB1 level and were increased in human samples with a better response to neoadjuvant chemotherapy and a better 5-year survival. So this suggests that the immune status differs between donors with increased HMGB1 levels in response to chemotherapy as compared to donors without increase, and further underlines the relevance of immune checkpoint marker monitoring to better adapt treatment strategies. Apart from the human preconfigured panels, mouse ProcartaPlex immuno-oncology checkpoint panel 1 and 2 enable the monitoring of soluble receptors and ligands to support studies on the response to potential therapeutic treatment also in mouse models, and have been validated by a collaborator in normal versus tumor tissues from mice. As demonstrated here in the two bar charts, analysis of in total 11 analytes in tumor tissue extracts from non-treated and treated mice revealed a distinct protein profile with differentially expressed proteins in the treated as compared to the control group, suggesting a role for these proteins in tumor progression. In addition to the immuno-oncology checkpoint biomarker panels, it is of great interest to analyze a broader set of immunomodulatory markers also in the context of immuno-oncology to get a more complete picture of an individual's immune status. So the human immune monitoring ProcartaPlex panel, which is actually the largest available panel on this multiplex platform, enables efficient immune response profiling, biomarker discovery and validation by analyzing 65 cytokine, chemokine, and growth factor targets simultaneously in a single well, and is complemented by an easy-to-use software solution to also enhance data analysis. So overall, ProcartaPlex immuno-oncology immunoassay kits allow simultaneous quantitation of up to 38 soluble immune checkpoint molecules for humans that are included in the immuno-oncology checkpoint panel 1, covering all the relevant soluble T-cell checkpoint molecules, while the immuno-oncology checkpoint panel 2 contains soluble natural killer cell checkpoints and are complemented by additional targets in the immuno-oncology checkpoint panel 3, which are now all combined in one immuno-oncology checkpoint panel that will be soon available to ultimately enable the systematic analysis of all these soluble biomarkers and to provide a more complete profile of cancer immunity. Besides, several immuno-oncology biomarkers for mouse are combined in the mouse immuno-oncology checkpoint panel 1 and 2, as mentioned, and we further continue to expand our portfolio of ProcartaPlex kits for cancer research, providing a cell proliferation and metastasis panel, in addition to available targets for angiogenesis or apoptosis, and there is also more coming soon to support this key research area. If there is still a target of interest which you can't find in our product portfolio, you can take advantage of our services, including contract development of immunoassays, which we provide for all available platforms, or our customer sample testing service. If you do not have access to an appropriate instrument or the capacity to run the assay by yourself, you can send us your samples and get your data without effort, and many other custom solutions that help shorten the time from sample to answer and to support your research. Finally, please feel free to visit also the new resource hub on our website that provides practical guidance and protocols and contains substantial technical content, including latest research tools, particularly for exploring the vast field of immunology. So with that, I will end this presentation, and I'm happy to take your questions now.
Thank you, Sigrun, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. Let's get started. Our first question is, what are the antibody response kinetics to SARS-CoV-2 proteins?
So that's a good question. Usually IgM and or IgA are among the first antibodies detectable within a few days upon SARS-CoV-2 infection, and thus indicative of a more recent infection, while IgG is a longer-lasting antibody, potentially also for monitoring of long-term immunity. However, the dynamics of the immune response and antibody response can vary depending on viral load and several other factors. And some individuals do not even produce detectable levels of antibodies due to several reasons. So in this case, further long-term studies are still required to better understand SARS-CoV-2-induced antibody responses and kinetics. In our portfolio of immunoassays for SARS-CoV-2 research, we are included IG total ELISA and ProcartaPlex multiplex kits for detection of total antibodies, as well as ELISA kits specific for detection of IgM or IgG. So you can choose the assay appropriate to your research need and depending on the research question that should be answered. As shown in the presentation, the ProcartaPlex 11-plex panel was also used to study total antibody responses to the 4 different SARS-CoV-2 antigens in ICU subjects over time. And more details on this study can also be looked up in the LabRoots webinar presented by Dr. Doug Fraser, which is still available on demand.
Thank you, Sigrun. Our next question is, in view of recent reports on SARS-CoV-2 mutations, does the ProcartaPlex multiplex panel detect SARS-CoV-2 variants?
That's a good question. So as with most viruses, mutations are common and they occur naturally and frequently, which means that variants are expected also for SARS-CoV-2. Since the human coronavirus IG total ProcartaPlex panel is a multi-target assay, so capturing antibodies to more than one SARS-CoV-2 specific antigen, so the broad epitope coverage, as mentioned, is an integral feature, and in this context and in this case, a clear strength. At this time, an impact on the overall assay performance is thus not expected, as convalescent samples, so from subjects previously infected with the SARS-CoV-2 variant, will still contain portions of antibodies directed against epitopes that are not affected by a certain mutation. However, it cannot be excluded that mutations might probably result in exposure of new epitopes or in masking of existing epitopes. So the question that remains to be answered is whether and how this might then change the immunoglobulin patterns identified using the ProcartaPlex multiplex panel. To date, we have no evidence for the impact of variants in this regard.
Thank you again, Sigrun. Our next question here is, do you provide any assays for detecting SARS-CoV-2 neutralizing antibodies?
So yes, indeed. A SARS-CoV-2 neutralizing antibody ELISA will be available very soon, which serves as a surrogate virus neutralization test, meaning that it will determine the neutralizing potential of antibodies in less than 2 hours without the use of live virus or cells, and can be performed outside of a biosafety level 3 environment as compared to conventional virus neutralization tests. So in addition to the information on the SARS-CoV-2 specific antibody titers that can be obtained by using our serological ELISAs or the multiplex ProcartaPlex panel, this ELISA will also allow to functionally characterize antibodies. So in terms of their neutralizing potency, and as pointed out in the presentation, functional neutralizing antibodies are considered effective in viral blockade and are thus indicative of potential protective immunity following SARS-CoV-2 exposure or vaccination. So this assay might also add value to studies on the immunological memory and that monitor also efficacy of vaccines.
All right, thank you again, Sigrun. And our last question for today is, can immuno-oncology checkpoint multiplex assays be combined with certain cytokine targets?
So yes, so apart from the pre-configured panels mentioned, it's possible to either blend assays for single targets of interest to create a multiplex panel, or use a mix and match offering to design a multiplex panel with the targets of your choice and desired species and sample type selected from the list of available analytes, assisted and using the panel configurator and selection tool on our website.
Thank you again, Sigrun, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.